Hi, everybody. I'm Jesse, Eurofins Discovery based in Taiwan, and I'm technical manager. Today, I want to present the future of carcinogenicity risk assessment. I will introduce some of the in vitro assays, including AI-powered ethical alternatives to traditional animal study. First, I want to thank you to everyone for attending this webinar. And today's presentation is based on the FDA guidance on carcinogenicity testing for pharmaceuticals. In November 2022, the FDA issued a new guidance, S1B Revision 1 Attendance, which introduced a weight of evidence approach to assess cancer risk in drug. This approach is part of FDA's efforts to reduce animal testing while maintaining safety requirements. For any drug expected to use more than 6 months, carcinogenicity risk study should be conducted. Traditionally, all carcinogen studies follow the guidance established by ICH, which have involved animal study to determine whether the substance can cause tumors. According to ICH and OECD guidance, the study involved hundreds of animals and normally 2-year study. Animal numbers typically consisted of 50 to 65 animals per sex per dose level, with 3 dose levels and double control groups at a cost exceeding $2 million. This is the most time and resource-consuming study for toxicology. However, in 2022, new guidance introduced a more refined weight of evidence approach. This new framework evaluates safety key categories, including drug target pharmacology, toxicology study, and specifically investigation. Based on this comprehensive assessment, we can now evaluate whether a traditional 2-year rat study would provide additional insight into understanding human carcinogenesis risk. The evaluation leads to 3 possible outcomes. You can see on the left side and the lower part of the slide: unlikely, likely, and uncertain. If the outcome is unlikely, evidence indicates that the rat study would not add value. A likely outcome also suggests a rat study would not add value. Only if the outcome is uncertain, you don't have enough evidence to make clear determination, meaning that a 2-year rat study could still provide valuable insights. Typically, the decision to waive this study depends entirely on the judgment of regulatory authorities in each country. For example, the US FDA, EMA, European Medicine Agency, and the other authorities will make decisions based on the specific quality features of the drugs, risk assessment results, and the available scientific evidence. This slide shows the 6 weight of evidence factors outlined in guidance for the pharmaceuticals: drug target biology and the primary pharmacology, including drug target distribution and the metabolite activity across species; secondary pharmacology screening that inform of target effects and the binding to receptor, particular carcinogenicity related targets; histopathology data for repeating dose toxicity study, particularly from 6 months animal study; and evidence for human hormone effect, including endocrine response mechanism, and the genetic toxicology data based on ICH S2 guidelines, including in vitro assays like AIMS test, micronuclear assays. And the last one is the immunomodulation according to ICH S8 guidelines, where the immunosuppression may indicate human risk for carcinogenicity. Today, Eurofins has specialized testing panels aligned with these factors. This is also the outline of today's presentation. Safety screening panels and the carcinogenicity risk assessment panels for secondary pharmacology. AI-assisted cell transformation as a phenotypic assay for histopathology in vitro assays. Endocrine system focused panel for hormone effect. And the in vitro genotoxicity test are well-known study for IND filing. And the last one, biomap for immunomodulation assessment. This integrated approach helps carcinogenesis risk assessment while supporting the 3R principle of reducing animal testing. The first factor is primary target biology and the pharmacological mechanism. Detailed text description of the guidelines on the slide for your reference. I don't discuss factor 1 in much detail, as it is the primary target that requires detailed investigation of its mechanism as well as carcinogenic risk by the drug developer. Secondary pharmacology is a crucial weight of evidence factor. It involves screening both the parent compound and its major metabolite to understanding their selectivity and off-target effect to carcinogenic risk factor. This off-target assessment operates on two key principles. First, establish a connection between molecular targets and the cancer-related pathway. Second, conduct in vitro testing against multiple off-targets to reduce toxicity risk. The main tool for this evaluation is cancer-related off-target panel. A publisher review article outlines an integrated strategy for non-genotoxic carcinogen. It highlights that exposure to certain chemicals can trigger various cell response related to carcinogenesis, like inflammation, immunoreaction, cell division, and cell injury. If the exposure continues, it can lead to further cell proliferation, transformation, and finally become tumor cells. Because of this, identifying the right molecular targets could serve as a useful model for assessing the risk. The review also indicates these targets, including a group of receptors and the enzymes. Here, I will introduce the carcinogenicity risk assessment panel focused on key targets involved in tumor formation, such as receptor binding assays and enzymatic assay for receptors, kinase, protease, CYPs, and more. Our target selection is based on systemic survey on PubMed literatures, emphasizing the most relevant receptor addressing carcinogenesis risk. Here is the result of our panel case study. We conduct screening profile across 30 different targets for various compounds, including some well-known carcinogens, non-genotoxic mutagen, and non-carcinogens. Our findings are really promising. You can see on the table. As shown in the heat map, different blue shows the strength of binding inhibition. The darker color indicates stronger interaction. What's special notable is that our panel identified all potential carcinogens, but not mutagen that work through directly genotoxic mechanism, like interacting with DNA not receptor or enzymes. At top of our chart, PMA, cyclosporin A, and triglyceride shows binding patterns across multiple targets. This multiple targets profile indicates compounds with potential carcinogenic risk. But compounds like cyclophosphamide, mitamycin, and aflatoxin, which directly interact with DNA and cause genotoxicity, could not be fully identified just by looking at receptor binding. This really shows why is it important to combine different assessment methods like genotoxicity study. In addition, the non-carcinogens, aspartame and methylparaben, showed little interaction with low risk. Combined receptor profiling with traditional genotoxicity tests give us a more complete in vitro system for assessing toxicity. This approach fits nicely with the guidance and to evaluate compounds in the early stage. Notably, our panel was cited in a 2024 review on Frontier in Toxicology Review, published by Benson et al., which focuses on the standardized of weight of evidence criteria. In the section discussing cancer-related off-target screening panels, highlighting Eurofins Discovery's panels to carcinogenicity assessment. Although the comprehensive list of risk targets is still being developed, Eurofins panel has gained significant attention, with many international clients, including those advised by the FDA. We know that we are making progress in the right direction. The third factor focuses on histopathological data. This including the finding from repeating dose toxicity study, with particular emphasis on 6 months rat study. But this is an animal study. Now, I will highlight the cell transformation assay, or CTA. The BHAS42 CTA is designed to predict carcinogenicity of compound with cell models and the phenotypic assays. This is significant because it is published in OECD guidance that can evaluate tumor-initiated or promote as an animal alternative method. We are using mouse BHAS42 fibroblast cell line, which transfect to have high carcinogenic potential for foci formation. This assay allows us to evaluate how the chemical affects cell growth and the transformation process. A key advance is that it can detect both genotoxic and non-genotoxic carcinogen. The endpoint measurements are 2 key parameters of cell transformation assay. First, cell number by staining directly measure cytotoxicity. Second, we assess morphology transformation by counting transformed foci, which indicates actually carcinogenic potential through visible cellular change. For the initial assay, we plate fewer cells per well in the plate, then apply chemical treatment during the cell growth phase. This specially detects compounds that may initiate carcinogenesis through DNA damage or mutation. For the promotion assays, it begins with higher cell density with chemical treatment during the stationary phase between day 7 to 14. This approach targets compounds that promote the cell growth of already transformed cells. The graph on the right of the slide displays the BHAS42 cell growth curve, showing the relationship between cell proliferation stage and the treatment windows. Note the log growth phase from day 0 to 4, followed by the plateau phase from day 6 and after. The black and the white bars below indicate when treatments are applied in each assay. This slide shows the example result of the conventional method for the cell transformation assay. As you can see, the assay measures carcinogenesis using non-positive control compounds. On the left, we have MCA for the initiation assay, and on the right, PMA for the promotion assay. We demonstrated the dose response effect of foci formation using MMNG in the initiation test and the 2AAF in the promotion test. The conventional methodology relies heavily on several manual processes: Jigsaw staining for visualization, focus counting technique, and critically, scientific judgment for interpretation. Here, I would like to introduce our new approach to improve cell transformation assay with AI-based image analysis. Compare the conventional and the advanced methodologies side by side. The conventional method presents several operational challenges. It requires 2 expert analysts working with 6 criteria per plate, per concentration, and per compounds. Despite its certification as a guidance document, the process remains labor-intensive, time-consuming, and the most important, subjective in its assessment. These limitations can impact both the throughput and the reproducibility. Our advanced method addresses these challenges through the implementation of AI-based in-kata analysis software paired with the image system shown in the photograph. This system has been extensively trained on more than 4,000 transformed foci, creating a robust foundation of consistent objective analysis. The dramatic efficiency improvement is quantified in the table at the bottom of the slide. For a standard analysis of 36 plates, the manual method typically requires between 17 to 18 hours of expert time. In contrast, our AI-based in-kata system completes the same analysis in just 8 to 9 hours, a nearly 90% reduction in the processing time. This advancement represents a big leap forward our histopathological assessment capability. By eliminating subjective variables while increasing throughput, we can deliver more consistent, reliable evaluation. Here are histopathological results comparing case study findings with the reference literatures. This slide showcases our examination of chemical carcinogens using the AI-assisted cell transformation assay. From the top of the table, we have known carcinogens. Benzopyrene demonstrated clear initiation activity in both our case study and the reference. And opiatic acid shows promotion activity aligned with the reference data. But we can see cyclosporin A and the ranitidine shows promotion activity in our study. No literature finds some difference. And MNNG and acetylaminofluorine demonstrated consistent alignment with reference data across both models. In the red row, you can see positive control chemicals performed as expected. MCA confirmed initiation capability, while PMA validated our promotion models. Both controls align with literature findings, reinforcing the validity of our approach. And in the green row, caffeine and mannitol, the negative control consistently showed no carcinogen kinetic activity across all evaluation. This result validate the AI-assisted cell transformation as a reliable method for objective assessment for carcinogenesis potential through both initiation model and the promotion model. We will keep improving the assay protocol to make it more robust and scientifically convincing. Moving to the number 4 factor, key data sources to form a systemical approach evaluates hormone effects. First, the repeated dose toxicity study, examining both gross and the microscopic changes in endocrine and reproductive organs, and showing potential hormone disruption. Second, reproductive toxicology study often reveal additional histopathological change relative to the endocrine function. Third, the compound's primary pharmacological mechanisms. Understanding whether a drug directly interacts with hormone pathway or receptors is crucial for predicting endocrine disruption. Fourth, secondary pharmacology of target activities might affect endocrine system. This unexpected interaction can sometimes be more concerning than the primary mechanism. As shown in the center of the slide, this data streams coverage to inform the hormone effect assessment. Here, we focus in vitro ways on nuclear hormone receptors, or NHR, which play a critical role in endocrine-related carcinogens. Multiple cancer types involve hormone disruption mechanism, like breast cancer, endometrial cancer, ovarian cancer, prostate cancer, testicular cancer, or other cancers. On the right side of the slide shows the physiological expression of nuclear receptors throughout the body. Green indicates tumor inhibiting receptor. Red shows tumor promotion receptor, and blue is with dual nature. This distribution helps us understanding why certain tissues are particularly affected by endocrine-mediated carcinogenesis. At the Eurofins, we have developed specialized screening panels to evaluate interaction with these critical targets. Our endocrine pharmacology panels, you can see on the left and the lower part of the slide, PP321 and the P402, evaluate nuclear hormone receptor and enzyme. This panel allows us to systemically screening compounds for potential interaction with endocrine targets, especially early in the drug development. Again, we have a case study on our nuclear hormone receptor panel. A detailed heat map showing how these compounds interact with various nuclear hormone receptors. The blue bars indicate inhibition effect, while orange shows activation effect. You can see cyclical and optical acid demonstrated strong effect on CAR and FXR respectively. These two compounds can bind into their primary target without off-target to other NHRs. The profiles like acetaminophen and methoxyamine C reveal some mutagens do not interact with nuclear receptor in the panel. Again, they interact with DNA. We need a genotoxicity evaluation. In contrast, low triglyceridone and is a PPAR gamma activator. It also modulates steroid reductase and the androgen receptor. The acetylstilbestrol also shows extensive modulation of estrogen receptor as well androgen receptor and the CAR, which explains its potential primary estrogen effects and is of target to carcinogenicity potential. And you can see bisphenol A, a compound of highly environmental concern, showing a diversity binding profile with estrogen and androgen activity, supporting concerns about its endocrine disruption potential. The last, methyl isothiazolene, as a cosmetic preservative, does not modulate these receptors. This binding profile provides crucial insight into how this compound may influence hormone signaling pathway. By understanding this molecular mechanism, we can better predict potential toxicology outcomes, carcinogenicity risk, and also evaluate environmental impacts. The genotoxicity testing is another critical factor. The testing method, including both in vitro and in vivo methods, as following OECD guidelines and listed on the table for your reference. For in vitro assays, we utilize the EMS test to detect DNA point mutation and the in vitro micronucleus test identifying chromosome damaging. Below are related assay number for your reference. As these in vitro genotoxicity studies are conventional requirement for IND submission and well known within the pharmaceutical industry, we will provide only a brief overview. Here we see the last point 6 on our slide, evidence of immunomodulation. The evidence on broader immunosuppression may provide high concerning for carcinogenicity risk. At Eurofins, we utilize the Biomap Diversity Plus, a human primary cell-based translational profiling platform to assess potential immunomodulation effects. The advanced platform features 12 systems with 148 total endpoints, allowing us to capture a broader range of human biology responses. The platform is organized into various key biological domains, such as vascular, fibrosis, immune, and respiratory system, or others. This approach effectively modulates adaptive response, like inflammation, immunoresponse, tissue remodeling, and the wound healing processes. The comprehensive approach allows for broader biological profiling and the toxicity signature identification, including immunomodulation potential and the associated risk. Biomap creates a phenotypic fingerprint of modulation, as you can see on the slide. Using human primary cell from 3 healthy donors cultured at low pH and stimulated with disease-relevant cytokines. This allows us to model complicated disease biology and measure the precise impacts of target agent on protein-based biomarkers. This slide is our case study testing cyclosporin A on Biomap platform. In the upper part is a phenotypic fingerprint across multiple cellular systems. Each column is a different cellular model. The colored lines show cyclosporin A's effect at different concentrations on various biomarkers. So specifically, the downward inhibition of cyclosporin A in the SAG and the BT systems. The pattern reveals cyclosporin A significantly immunosuppressive activity. The lower part of the slide shows immunosuppression clearly identified at very low concentration. You can see it is 40 nanomolar. This immunosuppressive effect directly connects to carcinogenesis through the impairment of immunomechanism, because the body's ability to recognize and eliminate neoplastic cell is reduced. This approach provides crucial early insights into potential immunomodulative effect that might contribute to carcinogenicity risk. This slide illustrates how we conclude integrated key weight of evidence factors to assess human carcinogenic risk according to the guidelines. This is adapted directly from the guidelines. The framework helps determine whether a 2-year rat study is more or less likely to be necessary. There are 6 criteria factors are evaluated, as we mentioned today: target biology, secondary pharmacology, histopathology, hormone effect, genotoxicity, and the immuno effect. For each factor, the assessment moves along a spectrum. To the right side shows less likely for 2-year animal study, meaning well-identified biology, highly selectivity, and no other concerning findings. And in contrast, left side shows a condition where traditional 2-year studies are more likely needed. Poor signal pathway, high off-target effects, or other highly concerning findings, or even unknown data of these 6 factors. This integrated approach informs decision-making about resource-intensive carcinogenicity testing. It is animal study. The last slide demonstrated how these weight of evidence factors are implanted across the entire drug development pipeline. The timeline shows how assessments involved. Our key innovation approach in today's presentation are conducted during lead identification and optimization stages. Target-based carcinogenicity panel for off-target screening, AI-based cell transformation phenotypic assay for histopathological study, genotoxicity study, especially in vitro studies, nuclear hormone receptor endocrine panels for hormone modulation or effects evaluation, and the biomap for immunomodulation screening. The last thing I want to emphasize for the conclusion is the integration of these factors showing advanced in carcinogenicity risk assessment. By combining mechanism understanding of receptor interaction, like carcinogenicity and the nuclear hormone receptor panels, then we can follow by AI-powered phenotypic assay like cell transformation assay or biomap. We can make a more informed decision about compound safety. Of course, it also supporting the 3R principle for animal testing reduction. Thank you for your attention. I'm happy to address any question about this integrated approach. Thank you.